We'll discuss now the antibody. The thing which you have to keep in your mind is that this antibody is a molecule. What it is? It is a molecule. You know that there is something called as the atom. If I put singles, this is called as the atom. When I put this and will join with one chemical bond, this is called as the molecules. So in the same way, antibody is like a molecules. It is a molecule. It is a precious molecules. It is a very, very precious molecules in your body. Fine. Now, this antibody, which is a molecules, now what is their chemical nature? The chemical nature is they are glycoproteins. Antibodies are, it is a glycoproteins. Fine. And we discussed in the previous classes that antibodies are, you can say, formed from, yes, B lymphocytes. Formed from B lymphocytes. So actually what is happening, from the B cells, B cells is formed from where? From bone marrow. From bone marrow. So from the B cells, after the maturation of this B cell, after the maturation and differentiation, the two things, maturation and full differentiations, the antibodies is formed. The antibodies are formed. Okay? Antibodies are usually Y-shaped. It is Y-shaped molecule. You got the point? From the B cell, after it get mature, differentiated, then antibody comes out. Antibody comes out. The first thing. Second thing is that in our blood, there is a two component. One component of our blood is the cells, different cells, RBC, WBC and platelets. In the other component of our blood is the plasma. You studied, no, in blood physiology, two component of blood. One is blood cells, also called as the blood corpuscles. Another one we have the plasma, the fluid part of the blood. Plasma constitutes 55%, this constitutes 45%. Now this 55% of the plasma, it is having the various things, and one of the things is plasma have the plasma proteins. Plasma have the plasma proteins. We have the three types of plasma protein. One is called as the fibrinogen, albumin and very good globulin. Fibrinogen, albumin and globulin. Fine. So these three category of proteins we have in our blood. These antibodies, they are formed, or you can say they are seen as a globulins. They are seen as a globulin. That's why it is also called as the immunoglobulins. So antibodies are also called as the immunoglobulins. Okay, come fast. Immunoglobulins, globins, or you can write globulins. Immunoglobulins. Understanding? No problem. So there was one scientist named Thales. What he did, he basically separated. He basically separated the entire three, you can say, plasma protein, okay, through one mechanism that is called as the electrophoresis. Have you heard about the electrophoresis? No? In the electrophoresis, what we are doing, we are putting the protein components and we are providing the electricity. Negative electrode, here is your positive electrode, and we run here the proteins. This is the one way to separate the molecules on the basis of charge, and basically on the basis of charge we are separating the molecules into a smaller size and larger size, smaller size and larger size. So when electricity runs, plasma, there are three plasma protein as we discussed. The one is called as the fibrinogen, another we have is the globulin, and we have the albumin. Okay, plasma proteins are negatively charged. Okay, plasma proteins are negatively charged. And the thing is, you know that if the protein is negatively charged, it will attract toward the positive charge. That's a simple, you can say, electrostatic rules. Like charge repel each other and unlike charge attract each other. So the thing is, when we put, you can say, the plasma protein into this electrophoretic, you can say, chamber, then what will happen? These proteins will start, to go through the positive electrode, and the thing is, this portion, this is the globulins, and this is your fibrinogen, and this is your albumin, So it comes out that the globulins forms the antibody, hence it is called as the immunoglobulins. Fine. There are further, you can say, deeper concepts. You just no need to, you can say, write on your notebook. You just understand. And the thing is, this globulins is itself 3 types. Okay, called as alpha globulins, gamma globulins, and beta globulins. Okay, three forms of globulins. Clear? No problem. Now, the another thing which you have to understand about the antibody is that it forms, if you're talking about the whole blood, then it forms the 20 to 30%. It constitutes the 20 to 30% of your blood. In 100% of the blood, the 20 to 30% is your antibody. Means you have the much more antibodies. The prime duty of antibody is to 2-3 things you just understand. The first thing is, antibodies are glycoproteins. Fine. You can say, recognize the antigen. It's recognize means पहचानता है first. It's recognize the antigen. Then it's bind with that antigen. And facilitate the clearance of antigen. Means their first prime duty is to recognize. Second duty is to bind. Third is to encounter or help in the encounter. Both the things. Either encounter by his own effort or by helping the other immune cells to encounter. Clear? Yes? Understandable? No? Fine. So this much you understand, and now let's move ahead and we discuss the structure of antibody. Okay? Structure of antibody. And one thing you remember, yes, through the antigen specificity, or you can say the binding affinity or binding style of antibodies, okay, based on the binding, you can say, specificity with the antigen, antibodies are classified into the 5 groups. Okay, it means antibodies are specific to the particular antigen. Okay, means one category of antibody will bind with the one separate category of antigen. Okay, then another category of antibody will bind with the another category of antigen. On this basis, we have the 5 antibody. The name you should remember by remembering one mnemonics, and that is called as the GAMED. Okay, IG, G, IG, A, IG, M, IgE, and IgD. This also helps you to remember their concentration. IgG is the most abundant antibody. It's a most abundant. Fine. Then second number is IgA. Then third is IgM. Okay. Then IgE and IgD. Got it? Yes. Now we'll discuss their structure. Till here, you have any question? No? in these days you do not have questions. I think you are like in hurry. You want to finish me fast. Yes? You want to finish this fast? No? Okay. Fine. So let's discuss about, that is the introductory part about the antibody. Now we'll discuss about the structure. The antibodies, they are tetrapeptide. They are tetrapeptide. Tetrapeptide means they have the 4 arm. What happened? Yes? Okay. Can go. So they are tetrapeptide. Listen here. Listen. Fine. So we are discussing the structure of antibody. Antibody structure is tetrapeptide. Structurally it is a tetrapeptide. Tetrapeptide means there will be a 4 peptide molecules. 4 peptide molecules. These 4 peptide molecules constitute the 2 light chain and 2 heavy chain. Can you recognize which is a light chain and which is a heavy chain? This one is a heavy chain. Very good. And this one is a light chain. The distinction made just because the heavy chains are more in weight, more than 50 Dalton in the weight. Fine. Talking about the light chain, it is less than 20 Dalton in weight, molecular weight. Molecular weight is measured in the unit of Dalton. Fine. So it is more than 50 and it is the less than 25 Dalton. So this distinction is made out of from their weight. Got it? No problem. Talking about the tetrapeptide. Why tetrapeptide? Because tetra means 4. Tetra means 4. So 2 heavy chain and 2 light chain. Fine. So we have the 2 heavy chain, okay, and 2 light chain. Now this 2 heavy chain and light chain, they must be joined. No, if there is a 4 types of the, you can say, molecules, they must be joined, otherwise they won't be, you can say, kept together for a longer time. So they are joined by a disulfide bond, disulfide means 2 sulfur bond, as like this. Okay, they are joined by a disulfide bond. Okay, they are joined by disulfide bond. Clear? Yes? Fine. Here also they are joined. Now the thing which you have to remember is that this antibodies prime duty is what? Its prime duty is to bind with the antigens. Now, what do you think? Where is the antigen binding site in this antibody? Where can antibody will bind? Here. No. Here. Turn. Here. Very good. Then I put it my finger here, you said no. Here. Very good. So the thing is, silence, the thing is, antigen bind with these areas. Antigen bind with this area between the, yes, heavy chain and light chain. Okay, these both are the heavy chain, don't confuse. Heavy chain and light chain, in between this, the antigen will bind. Fine. Now, these entire 4 chains, they will show, one part will show the constant, okay, another one will show the variability. Another shows the variability, means they will change their size and shape. They will change their size and shape. So this part, okay, this part, where you are? Where? What you are seeing? No, class was not interested. Huh? Then why your attention is not here? Okay, silence, focus. So we are discussing that here the antigen will bind. Fine. Then I said that there are certain area which can show the variability and there are certain area which is constant. So these area is constant. Okay, they will not change. But this area, it will change. It will change. So this is called as the constant region. This is called as the constant region. This is called as the variable region. Okay, this is the variable region and this is the constant region. Means in this heavy chain and in this light chain, one is the variable area. Okay, one is the constant area. Again, in the heavy chain, one is the variable region and one is the constant. And these both are the constant. Clear? Yes. Why it is variable? The reason is that all the antigens are not a same size, same shape and size. Some antibodies are oval in shape, some are this shape, some are this shape, okay, some are a square shape. Are you getting my point? Yes, sir. So, because there are so many types of antigen or so many different shape of antigen, yes? Yes? Yes, sir. Yes. So, this what is called as the antibody will also show the variability. Now, take this oval type and try to fit here. So, how you are going to fit? The thing is, in the antibody, there is a area where it bind. So suppose, So suppose this is your antigen, a bacterial antigen. Fine. Now, it will bind tightly. It will bind tightly. Antibodies basically bind tightly with the antigen. It bind tightly with the antigen. Okay. This binding, it is called as the affinity. It is called as the affinity. Affinity means binding. Affinity means binding. Fine. Now this area where it is binded, these are the most variable part. This is the most variable part. So this is also called as the hypervariable region. It is also called as the hypervariable region. Fine? No problem. And one antibody can bind with how many antigen? One antibody will bind with how many antigen? Huh? But it have the two arms. No, here also they can bind. No. Here bind. Here also bind. No. Yes, two. So one antibody will bind with the two antigen. Clear? Yes, sir. Fine. If there is a one antibody, this is called as the monomeric form. This is called as the monomeric or monomers. Okay? and it have the valency. It have the valency of how? 2. You can say the 2. It will bind with the 2 antigen. It will bind with the 2 antigen. So monomeric form of antibody will bind with the 2 antigen. So if you're talking about their valence, it is a divalent. It is a divalent. Are you confusing? Are you confusing? Let me summarize once again what we discussed. We discussed that antibodies are a glycoprotein. Chemically, they are a glycoprotein. It recognizes the antigen. It recognizes the antigen. Fine. It binds with the antigen. Fine. And facilitate their antigens clearance or make the antigens die. Clear? Now, then we've seen that antibodies is having the tetrapeptide structure. Tetrapeptide means 4 molecules. 2 is the heavy chain, bhari wala chain. 2 is heavy chain. Molecular weight is more than 50 Dalton. Then antibodies having the 2 light chain. Molecular weight is less than 25 Dalton. Then we've seen that in the structure of antibody, there is a specific binding area for the antigen. Fine. So the area where antigen is binding, this is called as the antigen binding site. Antigen binding site. In the antibodies, some areas are variable. They can change their shape. Some areas are constant. Some areas are constant, some areas are variable. And the specific area where antigen is binding, this is called as the hyper variable region, because here it have to bind, antibody have to adjust their shape so tightly that the antigen won't escape. Antigen भाग नहीं जाए, उसके लिए इस area of the antibody को बहुत tight होना पड़ेगा, बहुत variable होना पड़ेगा कि जिस तरह का antibody आए, I will change my shape. So this area is called as the hypervariable region. Then we discussed that if there is a one antibody is present, we are calling as a monomer, monomeric form. Are you getting my point? Then we said that there is a 2 antigen binding area in the one antibody. One antibody can bind with the 2 antigen. So it is called as the divalent. Valency. C. Valency means one antibody will bind with how much antigen? Valency means one antibody will bind with how much antigen? So one antibody will bind with the 2 antigen. It is called as the divalent. So this monomeric antibody will have divalent valency. Divalent valency. Fine. Talking about what is called as the affinity. Affinity means binding. Affinity means binding. Fine? No problem? Yes? Fine. Now, antibody can be monomeric form. It can be dimer form. Okay? It can be monomer. It can be dimer, means 2 antibody will bind, or it can be the pentamer, 5 antibody will bind. The 3 form of antibody we have: monomer, dimer, and pentamer. Fine? Means you drawn this structure? Shall I rub it? Yes? Fine. So the thing is, this is the one antibody, typical Y-shaped antibody. Fine. There is another antibody. Now, if it will bind, this is called as the joining component. J-shaped joining component. Fine. So if they will bind, this is called as the monovalent. This is the divalent. This is the dimeric form. Dimeric form, and the valency is no. This time no 2, 4. 1, 2, 3, 4. 4 area, 4 antigen can bind. So this is called as the valency will be the 4. Fine. Tetrameric. So valency is tetrameric, antibody is the dimer. In the same way, if there will be a suppose this is again the joining component, if the antibody will bind over here, change the color. Antibody will bind over here. How much? 3. Then antibody will bind over here. Fine. 4. Then antibody will bind over here. 5. This is called as the pentamer, pentameric form, and valency will be double, 10. You got it the point? Yes. So when we'll classify the antibody like Ig, IgA, IgD, IgE, and IgG, then when I will use the term that IgA is the monomer, then you have to understand it means there is a one antibody is present. If I will use the term it's a dimer, means there is a 2 antibodies present. Fine? Clear? No problem? So time is up. Time is up. Okay? We'll discuss the 5 classes of antibody along with their function tomorrow. Okay, and day after tomorrow we have another class for hypersensitivity reaction. So for concluding, you have any doubt, question? Yes? Binding. That is called affinity. Binding means affinity. Okay? We just clear the terms. I'll make you just write fast. It can be asked in the MCQ, so you just aware about this term. Yes. First one is the valency. Write it down. First one is your valency. Valency means antibody bind with how many antigen. As I said, you know, valency means antibody bind with how many antigen. Okay. Example you write. It can be monovalent, divalent, okay, or hexavalent, decavalent actually. Monovalent and divalent. Fine. Now, talking about the affinity. Affinity means, affinity means how together they are holding each other. We discussed, no, okay, there is something called as the antibodies present here. So how tightly they are holding each other, this denotes their affinity. This denotes their affinity. You can also write it down that the bond present between these antigen and antibody is your van der Waals force. The force which is present is van der Waals force. Then, which other force? Not electrostatic force. It's a van der Waals force as well as the hydrogen bond. Van der Waals force and hydrogen bond. Clear? Yes? Fine. Now, this is the valency, then affinity, then this is all. There is something more called as the avidity. You just write it down. Avidity. Avidity means how many point. Avidity means how many point antibody and antigen is interacting. Antibody and antigen is interacting. Clear? How many point? That denotes the avidity.
How many points antibodies
antigen will reacting. Okay? So you will see that there is something called as the agglutination reaction, clumping of the RBC, means pentameric form of the antibody. They will bind with the so many RBC and make the RBC to clamp. Okay, this is called as the agglutination reaction. We will see tomorrow. Okay. Till here, you have any question, doubt? No? Yes.
Antibodies में तो दो तरह के एंटीजन हो सकता है. वहाँ पे सेम ही एंटीजन हो सकता है.
क्या?
वहाँ पे सेम ही ये वो IgA जो एंटीजन है वो दोनों जगह पे सेम
Same ही, yeah. Very good question. Antigen which is going to bind with the two areas, they must be the same. ऐसा नहीं है कि इस पे लाइक ओवल शेप का एंटीजन आके बाइंड कर गया और इसमें रेक्टेंगुलर आके कर जाएगा. नो. जो फर्स्ट आर्म ऑफ द एंटीबॉडी में जो जिस तरह का होगा उसी तरह का दूसरा में भी फिट होगा.
शेप में तो वही होगा बट टाइप में भी वही होगा. IgG अगर पूछा है तो IgG ही पूछेगा.
No. Antigen is one, you cannot label as the IgA antigen. No. Antigen have the name according to the bacterial nomenclature. Okay? Clear? So let's conclude today's class. Basically, we discussed about the classification of acquired immunity, Yes. Then we started to discuss about the antibody. We discussed that antibody is the precious molecules. Why it is precious? Because it recognizes the foreign particles, the foreign antigen. It bind with the foreign particles or foreign antigen, and then it will kill or facilitate the clearance of antigen. Then we discussed that 25 to 23% of the constituents of the blood is formed by the antibody. Chemically, antibodies are glycoproteins having the tetrapeptide structure. In the tetrapeptide, two peptide is the heavy chain and 2 is the light chain. Between this heavy chain and the light chain, there is some area is called as the constant area and some area is the variable area. There is a specific site for the binding for the what? Antigen. That is called as the antigen binding site. One antibody is having the 2 antigen binding capacity. Fine? That is called as the valency. Then we discussed that antibody can be the monomer, it can be the dimer, or it can be the pentamer. Fine? We have the 5 classes of antibody. We can remember by remembering the mnemonics, and this mnemonics also provides us their concentration level. Concentration level. Starting with the Ig, gamed. G for the IgG. So G is having the more abundantly in our blood, in our body, the IgA antibody is present, and then slowly we have the less IgE antibody in our body. The thing which you have to keep in your mind is that a specific antibody will bind with the specific antigen. Not all the antibody will bind with all the antigen. No. One category of antibody will bind with the another category of specific category of antigen. Clear? In our tomorrow's class, next class, we will discuss about the 5 classes of antibody. So thank you for having me. Bye. Have a nice day.